Okay.
Okay, everybody, thanks for joining us today. It is an enormous privilege to have with us Dr.
Oh
Bob
gosh.
Dahlhausen. He is my
You're too kind.
go-to.
You're too kind.
Oh, well, you have been for years my go-to on this very complicated issue. And we
Thank you.
are beyond grateful that you're willing to tackle it in a way that more of us can understand it, especially, we work with a lot of veterinarians and I hear different stories everywhere and from people that have been to their veterinarian or they're trying to deal with their birds. So given that you have this unique patent on the test for this very complicated disease, I feel like you're right at the cutting edge of the knowledge, as you have always been, in my opinion. So I'm
And you're
go
too
off.
kind. Thank you.
Oh, not at all. I'm going to go off audio and video, and then we'll be back for what I'm sure is a robust Q&A at the end.
And if you have any questions throughout this talk, please, just, come on because, I'd like to address them at that point in time. Can you hear me okay? I hope so.
Yes. And people
Okay.
will put their Q&As in the Q&A, and then we'll try to consolidate those into, singularly helpful questions if needed.
Perfect. Thank you. Well, first, I would like to thank Ann and the whole staff at Phoenix Landing. These people are so wonderful, not only what they do for birds, but what they do to try to educate aviculturalists and veterinarians. I mean, hosting seminars like this. And, this whole topic of ganglioneuritis, I've got to give credit to my colleague, Dr. Susan Oros, who's about 4 hours away from me up in Toledo, and my colleague in Italy, who's a lot farther away, Dr. Giacomo Rossi, because I really think those two people, Susan's got a PhD in neurology. She totally understands this. She starts drawing all these pathways, and my mind kind of explodes when she does it, but she understands that. Giacomo, I mean, just what a brilliant man. I mean, he's light years above our understanding, the current researchers in that involved with Bornavirus, PDD, ganglioneuritis, and finally now they're starting to catch up with him. And this man, I'm 100% convinced, was totally correct 20 years ago. So with that, I'd like to talk about avian ganglioneuritis. I called it demystified. And why do I say that? Because there's a tremendous amount of misconceptions about avian bornavirus, PDD, avian ganglioneuritis. I mean, it's so often misunderstood. And it's not a simple virus equals disease relationship. We think, you get the flu virus, get the flu and recover. This whole topic is so different than that. And I think that's why we've got a lot of questions. So let's talk a little bit about, where it came from and, where we first seen it. Well, it's first described in the late 1970s in young macaws. These were birds that were imported from the Santa Cruz area of Bolivia. And it's really interesting because this was a period of time where we had increased importation for the pet trade of all these birds, these wild caught birds. Well, you know, did they bring the virus with them and transmit it to, say, cockatoos that came from Australia? Or were they already infected with the virus and, the Bornavirus and the stress of importation, did it cause it to manifest itself in a disease standpoint? And we're not sure about that. We really don't know. But the very first case was back around the early 80s. This was actually the very first macaw as reported in California. Oops, let me back up. And this macaw was wasting away. I mean, it would eat its food and pass whole seeds like you see here through the intestinal tract. And so they called it macaw wasting disease. And it was originally reported in macaws. And so that sounded okay. We're going to call it macaw wasting disease. However, we know now that in years to follow that it was reported in many other species of citizen birds. Not only did we see it in macaws, but we saw it in African gray parrots, cockatoos, Amazon parrots. We know birds like Quaker parakeets, lovebirds, and parakeets, budgies, are very resistant to getting this disease. And there was actually a study, I put this here, this was another topic. There's my pointer. There was a group at Texas A&M that infected lovebirds with this Bornavirus, and they didn't see any disease. And I can tell you from a clinician standpoint, I rarely see it in these species. I mean, I can probably count on one hand. So since we have it in these other species, macaw wasting disease was not a really good term for this disease entity that we were dealing with. And you'll hear this talked about synonymously with PDD and avian Bornavirus and ganglioneuritis. And I'm going to try to explain the differences here in this talk. So if you come away from this talk understanding anything, I hope you get this difference, okay? We know now that lesions were found in many other species of birds, canaries and finches, toucans, honey creepers. We know Canadian geese and even peregrine falcons. And actually, over 80 species of birds, both captive in aviaries and in the wild, have been identified with this. So macaw wasting disease is really not a good term for what we're dealing with. So what are we going to call it? Well, they looked at the lesions, and again, this is the first macaw that was reported, this blue and gold with this condition. They did a barium study. They saw the crop was dilated. This is a mature bird, and the proventriculus was huge, very, very big. So they called it proventricular dilatation disease. And that was a little more accurate. We thought, okay, we're going to call it PDD. Well, macaw wasting disease, we can't call it that. PDD, maybe, but we know now, and we saw with, as time went on, we could have lesions in the central nervous system causing ataxia, seizures, and blindness in these birds. We can have lesions in the autonomic nervous system affecting the heart and the adrenal gland. And I'll show this to you in a little bit later on here in this talk. And in the peripheral nervous system, the sensory nerves of the skin. And this is a common problem. This is the spinal cord, and this dorsal root, they call it the spinal cord, it kind of senses what the skin feels, and it becomes hypersensitive to stimuli. And so a lot of these birds will actually chew and mutilate, in these regions that this dorsal root, innervates. Um, so PDD really is not a very good term for what we're dealing with. Okay, so what do we call it? We can't call it macaw wasting disease. We can't call it proventricular dilatation disease. What are we going to call it? Well, this is a big question here. So what causes this problem? Well, back up a little bit here. There were a lot of different theories as proposed as to what causes it. All these different viruses. Paramyxovirus, that's pretty interesting, and I'll explain that in a minute. And then we know that in PDD cases where we had this abnormal digestive tract function, they also isolated adenoviruses, enteroviruses, and reoviruses. So these were all proposed years ago as being a cause of this condition. Well, it wasn't until 2008 where two independent reports, both by Kissler and Honkabori, and they use this little DNA chip here, and they correlated with Borna disease virus with cases of PDD. And I'll back up a minute here to talk about paramyxovirus, because before this time, before 2008, there was Dr. Grund in Germany developed a highly sensitive ELISA assay, and it was to the paramyxovirus that we see here. And sure enough, he could detect birds that had this disease. And based on that, back around 2000, we started running PCR assays for paramyxovirus. And what I did was I took the whole genome of paramyxovirus, I took conserved regions of it and mapped that out. And we had about 10 to 12 different primer sets for PCR. And we had one set that really had great correlations with cases of this disease. So I sent it out for sequencing, the product we amplified. And I was at the clinic, I was eating lunch, and I got the report back. It said Bornavirus. And as a veterinarian, I said, Bornavirus? I've never heard of this thing. This was around 2005, 2006. So I just kind of, you know, shoveled it away in the files and didn't think much about it. But then in 2008, they came out with this, and I'm thinking, wait a minute, we were amplifying a section of Bornavirus. And the interesting thing, going back to paramyxovirus is in the same family as avian Bornavirus is. And there was a paper that came out that showed the genetic composition of these two viruses. There's a lot of similarity between them. So when Dr. Grund was running this ELISA assay for paramyxovirus, and when we developed the PCR for this condition, we thought it was paramyxovirus, but indeed it was avian Bornavirus because they're very related viruses. So where did Bornavirus get its name? It actually came from a town in Germany called Borna. And back in 1885, they had an epidemic of a fatal neurologic disease in these cavalry horses. These horses became neurologic and they died. And so it was called Borna virus. And when they sequenced this virus from PDD cases, it matched in GenBank, which is its national repository of all these genetic sequences, Borna disease virus. So it was called avian Bornavirus. And the researchers here in the US, they cultured this from a confirmed case of PDD. They injected 3 healthy cockatiels with this avian Bornavirus, and 2 out of the 3 birds, 60 to 90 days later, developed clinical, quote, PDD. 3 out of 3 of the birds, when they sacrificed them, had pathological lesions very similar to cases of PDD. So these researchers concluded that avian Bornavirus is the cause of PDD. Well, they called it avian Bornaviral disease at that point. There was a big problem, though. And the problem was that we have cases of PDD that have been identified where they couldn't identify Bornavirus in the tissues. So I'm going to put this up here. This is a pathology slide, 2 slides. This is a process called in situ hybridization. And what that means is it stains the actual avian Bornavirus. Both of these birds are shown the infiltrate here that's compatible with PDD. This bird has the virus present here, but there is no virus in this here staining. But this bird's got the disease. So we can have PDD without the presence of avian Bornavirus. So we can't really call it macaw wasting. We can't call it proventricular dilatation disease. We can't call it avian Bornaviral disease because we can get disease without the presence of the virus. So what are we going to call it? Well, this is where my colleagues, Dr. Susan Oros and Giacomo Rossi, I mean, these people, they're amazing. Very, very intelligent individuals. And myself here, we decided to look at the disease process. In other words, what was the pathology that was occurring in these birds? And we decided this was an autoimmune disease that affected the nervous system. So we called it ganglioneuritis. So I'm going to back up a little bit and talk about gangliosides because this is important in understanding what's going on in this disease. So we're going to talk about the nervous system structure, okay? And in the nervous system, we have gangliosides, which form part of the membrane structure here of the nerve that we see here, okay? And there's over 50 different types of these gangliosides. And not only that, they vary in location throughout the nervous system. And as an aside, I'll tell you, this is a project we're looking at because we have birds that show central nervous system signs, peripheral nervous system signs, autonomic nervous system signs. And we don't know, is the autoimmune response to specific neurogangliosides that are located in specific sections throughout the bird, or are they mounting an immune response to a certain ganglioside that varies in its location among different species of birds? So I want to talk a little bit about avian Bornavirus, okay? It's a really interesting little virus because this virus, when it infects the bird, or in mammals, there's a mammalian Bornavirus, it has a propensity to infect the nerves. And this virus actually travels up the nerves to the nerve ganglia, which is the nucleus of the nerve cell. And they did studies in rats where they would inject it in the leg, but if they severed the sciatic nerve, the virus never got to the central nervous system. And once it gets to this nerve ganglia, generally the virus hangs out in the the nucleus of the nerve ganglia, but it also in birds will travel up to the central nervous system. So again, in mammalian species, if you cut the nerve, this virus can't get up to the central nervous system. And that's really interesting because we assume that, well, it's circulating in the blood, which it is, but it actually travels through the nerves to get up to the, to the central nervous system. And what does that mean in terms of birds here? And it means that the virus here can hide out in this cell nucleus. And that's very important because that's a very secure place for it to be. It does not stimulate an immune response, and it protects the virus from the host immune system hanging out in this nerve nucleus. And it's very similar to what we see with herpes infections. So that means that these infections are chronic, they're lifelong, and we don't get a very good immune response against this virus hanging out here in the nucleus. So persistent lifelong infections are common. I put this picture in here because we can see the nerve ganglia here, and this is a special stain for the avian Bornavirus. And you can see the Bornavirus here and how it's associated with these nerves. And that's how it travels basically through the body in mammals and in birds. Okay. So let's talk about gangliosides. Normally, these are isolated. They're compounds that are within this membrane of the nerve, and they're isolated from the host immune system. As the virus travels up the nerve, it exposes these gangliosides. They get exposed here as the virus is going through this nerve. And in doing so, then the host immune system recognizes them as foreign. Oh, we've never seen these before. We're going to mount an immune response to it. And that causes this immune response, which causes the disease signs that we see. As the virus goes up the nerves, it damages them and exposes these um, gangliosides. The immune response that we see then comes in and attacks that nerve and damages it. And that's very, very similar to what we see in some human diseases. We have immune cells, T cells and B cells. These are plasma cells. and they're sensitized to these nerve gangliosides, and they attack the nerve that we see here. We know that T cells, we call these killer T cells, and there's a whole group of them, but they directly attack the nerve that we see here. And then we have these B cells, which produce antibody that then combines, attacks the ganglioside or attacks the nerve. Then we get factors like complement macrophages that tend to degrade this nerve here. And so the disease that's occurring, the bird's own immune system mounts an immune response to its own nerve ganglioside proteins. This is an autoimmune disease that we're seeing that's occurring, and it's very similar to a condition in humans called Guillain-Barré, where we have antibodies that attach to and attack the normal nerve cell. Then we get complement macrophages and T cells that degrade that nerve cell, and we end up with damage to that nerve. And that's what's going on in these birds. And so when we look at a normal nerve, it innervates muscles, and we'll talk about PDD here, for instance. Um, if you take a muscle, okay, and you damage the nerve, this is a damaged nerve from this autoimmune reaction. That muscle, that corresponding muscle, that nerve innervates, will start to atrophy and degrade. And so we end up with, here's a normal proventriculus with a normal innervation. Here's a proventriculus whose nerve is damaged and the muscle starts to atrophy. Well, muscles make up the wall of the stomach of the bird, and they weaken. They start to disappear, and we end up with this dilation of the proventriculus. And you can see here, this is a normal radiograph of a bird with a normal proventriculus. And here's one where the proventriculus is dilated and enlarged in a case of avian ganglioneuritis. But we can also tend to see this in a crop. And here's a crop on a bird. And look how dilated and relaxed the muscles of that crop are in that picture. So what are we going to call it? Well, we look at the histopathology. We have sensitized lymphocytes, B cells and T cells that surround these nerves. This is a nerve ganglion cell, and these are all the infiltrated T cells and B cells, lymphocytes and plasma cells, and that causes damage to the nerve. So we call this ganglioneuritis. It's an inflammation of the nerve that innervates muscles and other parts of the central nervous system in the body of the bird. So we came up with this name ganglioneuritis, and I think it's very appropriate because it more appropriately defines the disease process that is occurring. We have, and if you send tissues from a bird that's positive to the pathologist, he will come back and report in these positive cases a lymphocytic plasmacytic ganglioneuritis. Lymphocytic, which are the T cells, the killer T cells, plasmacytic, the B cells that produce antibody. And it's an inflammation of the nerve ganglia, so it's a ganglioneuritis. And since it's in birds, we call it avian ganglioneuritis. And fortunately, this term now is starting to catch on, and even our colleagues that want to dismiss our theory here are starting to come around and call it avian ganglioneuritis. So what is it? Avian ganglioneuritis is an autoimmune disease induced by either avian Bornavirus invading the host nerve cells, okay? And these researchers had said avian Bornavirus is the cause of avian ganglioneuritis. However, we say it is a cause of avian ganglioneuritis because it's not the only cause. Bornavirus is not the only cause of this condition in pet birds. We can have disease without the presence of avian Bornavirus. And I put these two histopathology slides up here because we have the T cell and B cell infiltrate, which is quote, the ganglioneuritis. You can see all these little dots here. Those are TMB cells, and we can see it here. And these are both birds that are showing signs of, quote, PDD, ABV, ganglioneuritis, whatever. And these tissues were stained specifically for the virus. You can see the virus present in this bird. However, in this bird, there's no virus present. So we can have the exact same disease without the presence of avian Bornavirus. So this caused a real dilemma because the classic signs of PDD, AG, but these birds were ABV negative. So the researchers theorized, and Giacomo Rossi was the first, can we sensitize birds to these certain nerve gangliosides and cause, the same disease? So what they did was inject purified avian gangliosides into these cockatiels. They were either injected in the muscle or given orally. And they took 8 birds. These were the purified gangliosides. This was tested by PCR. There was no avian Bornavirus there. And 3 birds were given this orally. 3 birds were given it by injection. And 2 birds were just given a phosphate buffered saline as a control. Well, what happened? 1 month later, 100% of the injected birds and 1/3 of the oral birds developed central nervous system and gastrointestinal tract signs compatible with avian ganglioneuritis. 4 out of 6 of crop biopsies showed the same, what I termed lymphocytic plasmacytic infiltrate, which was compatible with PDD. So what they did was produce the ganglioneuritis, and you could see all the infiltrate of these T and B cells in this tissue from the proventriculus. They cause the disease without the presence of avian Bornavirus. And that's really important. Here's some pictures from those birds. You can see the enlarged proventriculus here in this cockatiel. And here's the uh, infiltrate that we saw in birds that were both injected with the gangliosides and given them orally to mount an immune response. Another group, 2 other groups actually demonstrated this, and one of the groups was actually at Texas A&M. But as I was mentioning to Ann earlier, they stopped the study right at the point where these birds were developing anti-ganglioside antibodies. And um, 2 of these other groups showed that these cockatiels that were negative for Bornavirus, but they were positive for avian ganglioneuritis, these birds that were inoculated to the gangliosides developed signs that were very compatible with PDD. This is a cockatiel that was sensitized, immunized against the gangliosides, then walled, ruptured proventriculus. And I can tell you from a clinician standpoint, I do not see cockatiels developing this disease normally. Okay. And that's, I'm going to back up a little bit here because a lot of these research papers that are out there use a cockatiel as a model. And this is a bird, a species that we normally don't see the disease in. And secondly, when they're infecting these birds with this large amount of Bornavirus, it's much, much greater than what they would normally be exposed to in a normal environment. So these studies really need to be questioned because I don't think they're accurately representative of what occurs normally in our birds. Okay. So again, the researchers that isolated the virus said that avian-borne virus is the cause of PDD. However, this is really not true because we demonstrated that, well, they demonstrated that it is a cause of PDD. That is true. However, it is not the only cause based on these studies where these cockatiels were inoculated and immunized against their neurogangliosides. Okay, so we're going to talk about one more aspect about this disease, and I hope this isn't confusing, but I'm going to explain a concept here because this is very important in understanding what can cause this condition in pet birds. And this is a condition called molecular mimicry. Okay. And what does that mean? That means that the host immune system responds to an infectious agent. This can be a bacteria or another virus. Okay. And that immune response, again, the T and the B cells cross react with certain nerve gangliosides, and that produces ganglioneuritis. Okay. So we know that we call this molecular mimicry. We know that it can cause ganglioneuritis. And we know that this inflammation, this cross reaction to the nerves or the gangliosides throughout the nervous system. And again, remember, there's over 50 of these different gangliosides, and they're located all throughout in different regions of the nervous system. Okay. So as the host immune system causes this nerve damage by cross-reacting with these certain gangliosides, even though it's sensitized to a certain bacteria or another virus like paramyxovirus, that sensitized immune system, because they're so similar, can cross-react with certain nerve gangliosides and cause the disease. So this leads to our commandment about avian ganglioneuritis. This is postulated by Susan, Giacomo, and myself. If it is true that avian bornavirus infection is a cause of PDD, it is equally true that PDD is not caused. It's not equally true that PDD is caused only by ABV. So, in other words, even though ABV can cause it, other things can also cause it. And that's very important. And so the ganglioneuritis, any infection that exposes the nerve gangliosides to the host immune system and elicits a suitable immune response, can cause disease that is indistinguishable from PDD, avian bornoviral disease, avian ganglioneuritis. Okay. So what we're seeing here about ABV infection is just the tip of the iceberg, because we can either get direct damage to the nerves, indirect damage through molecular mimicry. It's an autoimmune disease. We end up with avian ganglioneuritis. This antigenic mimicry is another problem because we know Campylobacter is a big one, which is a bacteria, the gastrointestinal tract, chlamydia, mycoplasma, herpes viruses. They all can sensitize the immune system against the nerve gangliosides and cause viral damage to the nerves. So that's the tip of the iceberg there. So gangliosides involved in different infections. We know that mycoplasma, if we get an immune reaction to mycoplasma infections, it reacts with these two gangliosides. And GM1 is one of the big ones we see in cases of PDD. Haemophilus paramyxovirus, also very similar to what we see with Bornavirus. But also these other viruses, herpes virus, Campylobacter, which is a bacterial infection in the intestinal tract. Certain strains will sensitize the immune system against the GM1 and GT1A gangliosides, which are very common in cases of PDD. So we can have other organisms that induce an immune response that cross-reacts with certain gangliosides and causes avian ganglioneuritis. So Campylobacter in the GI tract is probably one of the most common bacteria. We get an immune response. We get T cells and B cells that react. They produce sensitized killer T cells, and the B cells produce antibody, which goes out and circulates, but it cross-reacts with nerve gangliosides and causes nerve damage. And that's how this infection in the intestinal tract can cause signs that are exactly similar to cases of PDD or ABV disease. It causes a ganglioneuritis. We know these antibodies, again, they cross-react. They actually bind here to certain regions of the nerve here. And then that we get this complex where we get these macrophages, we get this is a cross-reactive antibody, we get sensitized macrophages, activates complement, and all these things end up damaging the nerve, and we get this ganglioneuritis. So this is a cross-reactive immune response that occurs in these birds. So let's talk about avian ganglioneuritis, PDD, ABV disease. In summary, right now, there are 3 ways that we can produce this disease. It's an autoimmune disease. So we know with Bornavirus, avian Bornavirus causes a limited pathology directly, okay? In other words, it doesn't really kill cells. It hangs out. They call it a non-cytopathic virus. And what that means when they culture it in these cell lines in the laboratory, it doesn't kill the cells. It just hangs out in them and it invades the host immune system, like I showed you in that image. It hangs out in the nucleus. It evades the host immune response. And it doesn't really kill the cell because that's where it's living. So the virus, as it transits through these nerves, it can expose certain gangliosides that the host immune system recognizes. And so the host immune response causes nerve damage, which we term ganglioneuritis, okay? That's one way we can get the disease. Other infectious agents, and I talked about paramyxovirus and herpesvirus and even chlamydia can expose certain nerve gangliosides and cause an autoimmune ganglioneuritis, just like avian Bornavirus. And then the other way we can get this condition is that the host responds to another infection that cross-reacts with certain gangliosides and causes damage to the nerves and disease, which is identical to PDD, ABV disease, or avian ganglioneuritis. And that concept is called molecular mimicry. So we have 3 ways that we can get avian ganglioneuritis or PDD. All 3 of these have been proven to be a factor in causing this disease. They all can cause it. Okay, so we know then we're going to change this from the original researchers that found ABV associated with cases of PDD, I'm sorry. And so we can say that ABV, avian Bornavirus, is a cause of avian ganglioneuritis, but it is not the only cause of avian ganglioneuritis. This is just an illustration of the nerve ganglia and all these little highlighted spots here. These light areas are that sensitize B and C cells that are attacking this nerve ganglia. So it's a ganglioneuritis that's occurring. Okay, so let's talk a little bit about the disease. The disease that we observe in birds is real variable. And it depends not only on the host species that's involved. Are these old world birds like cockatoos and African greys? Are they new world species like macaws and conures or African gray parrots? And also, what we see in terms of this disease depends on the distribution of these lesions. Are they occurring in the central nervous system, the autonomic nervous system, or the peripheral nervous system? And we know that there's some generalities that'll occur with this, okay? Um, Also, what we see clinically depends on the severity of the lesions, okay, which is really variable. Sometimes the signs are real mild, sometimes they're really severe. We know that old world species like cockatoos, we tend to see central nervous system signs. However, if you run pathology, you'll find lesions in the gastrointestinal tract like we do in New World species like macaws and conures. We'll tend to see signs in the autonomic nervous system, which innervates the gastrointestinal tract. African greys, we see signs in the central nervous system and the peripheral nervous system. And I'm going to show you a few examples of these, okay? So the classic disease that we see, which was identified in New World species in the blue and gold macaw and the scarlet macaw here, affects the gastrointestinal tract. And I talked about the normal nerves innervating the muscle. When these nerves are damaged, the muscle it weakens, it atrophies. And when that happens, we get this dilatation here of the proventriculus, and we see this bird cannot really digest its foodstuffs. The gizzard, the ventriculus, is how the bird actually chews its food. And when that muscle gets really weak, the seeds that the bird ingests pass throughout the GI tract here, throughout the stomach, the ventriculus, and go throughout the intestinal tract. And we see birds passing whole seeds in their droppings because this nerve that innervates the proventriculus in the upper GI tract has been damaged and the muscles basically waste away. Okay, so that's very common. Other problems with, and that's the autonomic nervous system. Other problems, we'll see the autonomic nervous system innervates the heart. So you can see problems with arrhythmias. This was an African gray that came in to us, was actually picking right over this region of the heart here on its chest. This bird, we didn't recognize, this was really early on before we knew about avian ganglioneuritis, but subsequently, when we ran the histopathology, this bird fell off its perch and literally died of what we call, quote, a heart attack. When we ran the pathology, it showed the characteristic infiltrates in the muscle of the heart, lymphocytic, plasmacytic infiltrate of the heart. This bird had an autoimmune disease or an avian ganglioneuritis of the nerves innervating the heart muscle. And I have to assume that that's why this bird started picking over this region here of the heart. Okay. We'll talk a little bit about ganglioneuritis in the central nervous system, and we'll start with the upper brain, the cerebrum, which is right here in this bird, okay? And this was a real interesting case that my colleague, Dr. Susan Oros, had in an Amazon parrot. And this bird developed what we termed cortical blindness. In other words, the eyes, when we looked and shined a light in the eyes, the iris would constrict. So the retina was receiving the light and sending a signal back to close this muscle down. However, when this bird was blind, because when it tried to form an image, a visual image, it couldn't form the image here in the upper brainstem. So this was central nervous system signs in this bird. And although the eyes were working and the retina was working, it wasn't seeing, quote, seeing the image in the upper brain of this bird. And this is the Amazon parrot that happened in. And Dr. Oros found that you had to increase the dose of treatment of Celebrex upwards to 60 milligrams per kilogram. And in treating this bird with that, this bird regained its sight. So this was avian ganglioneuritis of the central nervous system. Okay. We also see central nervous system signs affecting the cerebellum. I got to go on here. Okay. And we see this in cockatoos. These birds will show seizures. We do see it in some New World birds, some macaws, especially many macaws. And we get seizure activity up here in the upper brain in the cerebral hemisphere. Again, this is very common in cockatoos and African gray parrots. And this is a picture, and again, I mentioned many macaws. This bird is showing a focal seizure. The bird is conscious, okay, but it has this uncontrollable twitching of its foot. That is a very focal seizure that this bird had. And with treatment, this bird actually responded and this sign went away. Okay. We can see generalized seizures. This is an African gray showing that. Okay, so that bird was showing a little bit more than a focal seizure. It was a generalized seizure, but this bird still was conscious as to what was occurring around it. So a little more advanced than a focal seizure. And compare this to this cockatoo with a generalized seizure, which this bird just consistently, constantly just was twitching and shaking like this. This bird really wouldn't eat, wouldn't drink. It just had these movements and wouldn't do anything else. That's a more generalized seizure. So this is avian ganglioneuritis of the central nervous system. Okay. So another problem that we see, and this is really common with avian ganglioneuritis of the CNS, the central nervous system, is problems affecting the lower brain stem or the cerebellum. And this causes problems with ataxia, incoordination. These birds act like they're wobbly. They really don't know where they are. They're kind of, quote, drunk acting, so to speak. We also see these proprioceptive and motor deficits. And what that means is the bird doesn't know where its body is in three-dimensional space. And by that, I mean, if you close your eyes and say, take your finger and point it to your nose, you can do that without visually seeing it. That is proprioception. You know where your finger is in three-dimensional space, and you know where your nose is, and you can coordinate combining the two of them. Okay. Well, birds that have avian ganglioneuritis in this lower brainstem cannot do that very well. And we see this very commonly in both cockatoos and African gray parrots. Now, this is a young umbrella cockatoo that's showing this ataxia and incoordination. And you can see the bird shaking, showing a little generalized signs of seizure activity. The bird's conscious, it knows what's going on around it, but it's standing and it really can't catch its balance. It actually throws its wing out here to try to catch its balance. See how that wing is twitching and being thrown out because it's losing its balance. Even though it's not moving, it's standing on 2 feet. The bird really cannot, catch its balance very well. This is a bird that Dr. Oros had, an African gray that's showing these proprioceptive deficits. And you can see here what they're doing is they're pulling the wing out on this bird, and the bird will actually leave the wing outstretched because normally if you do that on a bird, it's going to pull the wing right back into its body. Now watch here, you'll see as she pulls the wing out, it kind of leaves the wing extended. Not so much, I think, on this left wing. It's kind of pulling it back slowly. I think the right wing here was much worse. You can see bird doesn't know where its wing is in three-dimensional space, and it just leaves it out there. And that's a sign of lower brainstem disease or ganglioneuritis in the central nervous system. Here's a little mini macaw again. And you can see that this bird wants, I'm sorry, I don't know if I can get the volume turned off on that, but you'll see how this bird wants to move, but it's got to use its beak because it's really insecure about its feet, moving its feet here. See, she's very unsure of herself. That bird is trying to move, but she's got to use her beak to help her out. This is another bird here that Dr. Oros had. And watch how this bird walks. It's trying to move, but it's really having difficulty here showing, with its feet, not knowing where they're positioned in, quote, three-dimensional space. Watch how you can see how this bird is wobbly. It's trying to walk, but it's really having a difficult time. And that's an avian ganglioneuritis again in the lower brainstem. Okay. We can also see ganglioneuritis in the peripheral nervous system. And this often occurs in the spinal cord here in the thoraco-lumbar vertebrae, okay? And this is a cross-section of the spinal cord. This dorsal root is the sensory nerves, okay? These are nerves that sense the skin and that of the feet and the chest and so forth. And there was a researcher that actually showed that birds had lesions in this dorsal root of the spinal cord who were feather picking, mutilating at the level of the skin, and also mutilating their skin. Okay. So this peripheral neuritis in birds contributes to feather destructive behavior and self-mutilation disorders. There's a little cockatoo that had this condition, and this bird would just, you can see all the scabs, this bird would just constantly pick and chew at its skin because it was hypersensitive to that skin because of the lesions that were occurring in the spinal cord. Here's an African gray parrot, and you can see the feet and legs here, and you can see how this bird was, again, hypersensitive to these areas of the skin and actually chewing on those regions. So this is what we term a peripheral neuritis. It's avian ganglioneuritis of the sensory nerves of the spinal cord. So basically, when we see disease, avian ganglioneuritis must be considered in any clinical disease that is neurological or neurogenic in nature, with or without the presence of avian Bornavirus, okay? So again, avian Bornavirus is a cause of PDD. It is not the only cause. And I'll talk a little bit about Bornavirus because this is kind of interesting. It's an RNA virus, and what does that mean? It's RNA breaks down. There's enzymes that break down RNA everywhere in the environment. This family of viruses that Bornavirus is in also includes paramyxovirus and rabies virus. So these are very neurotropic viruses that attack the nerves. And again, I mentioned that paramyxovirus and Bornavirus share certain parts of their genetic sequence that's identical. They're identical to each other. And that's why we can see infections with paramyxo that cause avian ganglioneuritis without Bornavirus. It's rare, but it does occur. ABV reproduces in the cell nucleus. And what does that mean? It evades the host immune system. So we'll tend to see asymptomatic infections and persistent infections, very similar to herpes virus. Once a bird's got avian Bornavirus, they've got it for their lifetime. And they will not, at least as far as we know to date, they will not get rid of it. These viruses are also very highly neurotropic. They affect the central nervous system, autonomic nervous system, and peripheral nervous system, as I explained earlier, showed earlier. ABV, there's over 17 genotypes of these right now. They affect citizen birds, passerines, canaries, finches, a lot of water birds, ducks and geese, and there's a bunch of unclassified Bornaviruses. So the bottom line is Bornavirus is widely distributed both in captive and in wild populations of birds. Michael Litz in Germany took 59 healthy birds, tested them for Bornavirus. These birds were clinically normal. 45% were positive for ABV. There was an aviary that had known cases of PDD. They tested those birds. 35 out of 77, or 45%, were positive for ABV. We tested 791 samples that came into our laboratory for other testing, and we ran them for Bornavirus. 271, or 34% of those, were positive for avian Bornavirus. So what does that mean? Well, there was a wide-scale survey in aviaries in Europe, 1,400 birds, 23% were positive. There's a similar infection rate in canaries and also wild ducks and geese, both here in the U.S. and South America. There's a similar rate of infection in wild citizen birds in South America. So we know the infection rate for avian-borne virus varies between 23 to 45%, an average of 30%. So 1 out of 3 birds is going to be positive on the average for avian-borne virus, okay? So Avian Bornavirus is present in almost every single aviary that we have here in the US and in Europe. And, um, if you've got more than 3 birds, chances are you've got Bornavirus in that collection. However, this is where we really get into this problem with testing and so forth, and I'll explain that in a minute. But most birds that have avian Bornavirus are normal, and most of them will remain normal throughout their lifetime. And I would say greater than 90+% of those birds are going to be normal. Okay. Avian Bornavirus is a very fragile virus. Its RNA, it rapidly breaks down when it's outside of the host body. Okay. At best, it'll last 5 to 6 hours under ideal environmental conditions. Okay. We know that the virus is shed intermittently in the urine and the droppings, but bird-to-bird transmission is extremely difficult. And there were several studies out there where they took Bornavirus, they cultured it, they gave it to cockatiels orally to simulate ingestion or eating the virus, or intranasally to simulate inhaling the virus. Not a single one of those birds converted to a positive viral status. Okay. So this virus is not contagious, really, under normal routes of exposure. Um, ABV is not easily transmitted from bird to bird. It may occur, but it's extremely rare, requires birds that are in close contact, in other words, in the same cage, a small cage, over a long period of time, and we're talking years, then it may be transmitted. However, it's extremely rare. There is one report, and this is only one report, where infection spread rapidly through a citizen nursery. They were hand feeding African gray parrot babies. These birds were just in the pin feather stage, so they were not immunocompetent. In other words, they didn't have fully developed immune systems at that time. And those birds, I'll back up a minute here, those birds within about 3 weeks developed neurologic signs. A lot of them died from it. We do not know if those birds were infected with the virus before they came into the nursery or if they acquired it from exposure within the nursery. This is only one case report that's ever been reported on this, okay? So how is Bornavirus acquired? How do these birds get this infection? Well, I remember in 2007, Dr. Helga Gerlach, who just a really brilliant avian pathologist in Germany, was speaking to Dr. Oros and myself, and she said, PDD is transmitted through the vitellin membrane of the egg. Well, we had a situation at the zoo where we had neurologic signs and deaths in chicks, which was compatible with avian ganglioneuritis. And in doing some testing, we found that both parents were positive for avian Bornavirus. And when we tested the vitellin membrane of the eggs, 22 out of 23 of those eggs were positive for avian Bornavirus. So what that told us was that most likely these Kia chicks were hatched out with already the Bornavirus present in their system. So vertical transmission through the egg right now is considered one of the most likely ways that birds acquire the virus. In other words, once they hatch out, they've already got the virus in their system, and there's nothing that you're going to do to eliminate the virus. They're going to have it for their lifetime. Again, you've got to understand that most positive birds are normal and will remain normal throughout their lifetime. Okay. So we know that Bornavirus is present in healthy birds. These birds don't show signs of clinical disease. We know these infections are lifelong. And the majority of these birds, again, I can't emphasize this enough, are normal and will remain clinically normal throughout their lifetime. And that's very important. And I've got to say, it's well over 90+% of these birds We know that factors like stress, especially reproductive stress, when birds come into breeding condition. I had one cockatoo that relapsed with ganglioneuritis 3 consecutive years during the breeding season. Nutritional stress, environmental stress, moving birds, birds like we had where we captured birds in the wild, moved them into quarantine stations, sent them off to pet stores and then into homes. That type of stress can all cause this virus to activate. Stress from other diseases can all activate avian Bornavirus and cause avian ganglioneuritis to occur. So the bottom line is in these positive birds, you want to maintain a very neutral, very calm environment, good nutrition, minimize the stress, and these birds will be fine. And that goes then to ABV testing, avian ganglioneuritis diagnosis. I mean, this is a real Pandora's box. And the reason I say that is because so many people think I've got a bird that's ABV positive. This is a fatal disease. My bird is going to waste away. It's going to develop PDD, waste away. It's very contagious. All my birds in my home are going to get this and they're going to die from it. And that is so not true. Okay. We know that the birds that test positive, again, the vast majority are clinically normal and will remain normal throughout their lifetime. So I'm going to talk a little bit about avian Bornavirus testing. There's a PCR assay for avian Bornavirus. The problem with this is there's no standardization among different laboratories, and we've got a number of labs here in the US that test for it. The test that most laboratories do does not detect all the genotypes of avian Bornavirus, okay? And it's very difficult. In fact, it used to be we had to run 3 different PCR assays on one sample to determine that that sample was indeed negative for Bornavirus, okay? We've got one set of very conserved primers that seem to work in all the cases. We're not certain 100%, but it's a very reliable assay. Most of the labs do not use that assay. The other problem with this testing is that Bornavirus is intermittently shed, excuse me, in the stool, okay? So if you're running fecal samples, the chances of you finding it, I'm sorry. Oh, okay. I didn't hear that, but I don't know what somebody said there. So we know that Bornavirus is intermittently shed in the stool. If you run a sample when it's not being shed, you're going to get a negative test result. So problems with testing are that most labs don't get all the genotypes. The virus may not be present in the sample you send in. Alternatively, it's shed at very low levels when it's there. And if the lab doesn't have a real sensitive assay, they are not going to detect it. Also, the virus degrades very rapidly in the samples that are tested. And we have shown this. We know that samples that are highly positive today, 7 days later, can test negative. So the virus, again, being an RNA virus, it breaks down real rapidly in the environment. So bottom line is a negative test, PCR assay for avian Bornavirus does not mean that the bird does not have the virus. It only means that the virus was not detected in that sample, okay? Again, positive samples degrade rapidly, usually within 5 to 7 days. So false negative test results are very common. False positives, not very common at a reliable laboratory. If you get a positive test, it's pretty reliable because not much is going to cause a positive reaction on a valid PCR assay. So again, the majority of ABV positive birds are disease-free. They'll remain normal throughout their lifetime. I keep repeating this because you've got to understand this. I get so many clients out here, my bird's got PDD, it's going to die. All my other birds are going to get it. And that is totally not true. It is not a highly contagious virus via normal routes of exposure. And I talked about those studies where they took active cultured virus and they gavage fed it into birds to simulate ingestion, and they dropped it in their nostrils to simulate inhalation. Not a single one of those birds became positive with the virus, okay? Other testing that's out there, there's a serology for avian Bornavirus. And what this does is it tests for antibodies against avian Bornavirus. The problem with this is the virus travels up those nerves and it hangs out in the nucleus of the nerve cell, and that is isolated from the nervous system. So you're going to get a lot of birds that have Bornavirus and are going to be negative on the serology, okay? And this has been proven where they've done PCR and found birds that are shedding Bornavirus, but the ABV serology was not positive. Not all birds shedding avian Bornavirus are serologically positive, okay? And then also, a negative avian Bornavirus serology doesn't mean that the bird doesn't have the virus. It just means the bird didn't mount a successful immune response to detect the virus and produce activated B cells that produce antibody that's detectable on this assay. So, and this is a test that is promoted out there to test birds for Bornavirus, okay? But it doesn't mean that a negative test doesn't mean the bird has it. Positive test doesn't mean that the bird has the virus. The bird could have been exposed to the virus, mounted an immune response that knocked the virus out. It never infected the bird, and that bird can be negative for the virus. So, bottom line on avian Bornavirus serology, it really is questionable. It doesn't really give us solid information that we can base decisions on, okay? It is not a real informative test. And you're going to see this test being promoted out there in the internet and that. And these are the problems with that assay, okay? I'll talk about avian ganglioneuritis diagnosis, okay? And this is the anti-ganglioside antibody assay. This is a serology test like the Bornavirus antibody assay, but this actually tests for anti-ganglioside antibodies. In this chart, I tried to simplify it. So what it shows here in this column are birds that have lesions that have been identified by the pathologist as positive for avian ganglioneuritis or PDD, okay? This column is birds that were positive on fecal swab for Bornavirus by PCR. This column demonstrates birds that have antibody to Bornavirus, and this column demonstrates birds that have anti-ganglioside antibodies. So here we have 4 birds in this group that showed classic signs of PDD. 4 of them were Bornavirus positive on PCR. Only 1 had antibodies to Bornavirus, but 4 of them had anti-ganglioside antibodies. Here we have a group of birds that did not have any lesions on the histology that were proof of avian ganglioneuritis. 4 of these birds were positive on ABV for avian Bornavirus and PCR. 4 were positive on the assay, okay? None of them were positive on the AGA assay, the ganglioside assay. So none of the birds that had 0 evidence of ganglioneuritis on their pathology, also none of those birds had AGA positive, but they were avian Bornavirus positive and they had avian Bornavirus in their stool. Here there were 5 birds that did not have the lesions. None of these birds were Bornavirus positive on PCR, but 5 out of 5 were positive on the antibody assay, okay? But none of them were positive on the anti-ganglioside assay. And then here we had birds that were negative for the pathology. 8 of those birds were positive on PCR for Bornavirus on the fecal sample. None of them had antibody for Bornavirus, and none of them had AGA antibodies. So bottom line is AGA is very, very indicative of the pathology that's occurring. And in cases where there's no pathology, the AGA was negative, even though you could pick up Bornavirus in some of these birds on PCR, and some of them were avian Bornavirus antibody positive, some were negative, some of them were positive without presence of the virus on PCR. So the bottom line in looking at all these three tests for avian ganglioneuritis diagnosis is that the avian ganglioside antibody assay is the most predictive of pathology due to avian ganglioneuritis. So in a study that Giacomo Rossi did, 98% of the birds with clinical disease had elevated AGA levels regardless of the presence of avian Bornavirus, that's PCR positive or negative on the fecal swab. So the AGA assay most accurately detects birds that are clinically diseased or that are showing signs neurologic disease due to avian ganglioneuritis. And that's really important. The ABV antibody assay is variable, not very informative. The ABV PCR is variable among laboratories. Positive result is generally reliable at a reliable lab. If positive, it does not mean that the bird has disease. If negative, all it means is that the lab did not pick up the virus in the test sample. It doesn't mean that the bird doesn't have it. The AGA assay most accurately correlates with birds that have clinically diseased ganglioneuritis, birds that are clinically diseased, okay? So I'm going to finish up a little bit here with treatment, and then we'll talk about, I guess we'll address some questions, and Ann was telling me she's got quite a few questions. Okay, so back in 2000, before we knew what caused PDD, ganglioneuritis, whatever you want to call it, we knew that it was an inflammation in the nerve ganglia. And I had a client who had lost birds from this. We had histopathology to confirm it. And he had a real loved pet blue and gold. This bird was his life. I mean, he just loved this bird. Bird started showing clinical signs, and he came into me, and I told him, I said, John, I said, you've been through this before. You know what we're dealing with. And he said, Doc, you've got to do something to save this bird. He said, this bird is my, all my life. He said, I really love this bird. You got to save it. So I'm thinking, what am I going to do? Because we don't know what's causing this problem. This is back in 2000, okay? So we don't know what's causing this problem. And so I looked at what are we dealing with? We have inflammation of the nerve ganglia. So I started looking and saying, well, how do we suppress that inflammation? And I thought, well, the steroid use in birds is pretty contraindicated. It tends to suppress their immune system. You can use short-acting steroids, but we can't do this for a long period of time. So I started looking at non-steroidal anti-inflammatory agents, and in evaluating all of them, I came across Celebrex. And this was really interesting because it had really high bioavailability, which what that means is when you give it orally, a large portion of it is absorbed from the gastrointestinal tract. It tended to concentrate in the gastrointestinal tract, okay, which was good. And it's what we term a specific COX-2 inhibitor. Okay, so what this means is there's two types of this enzyme, this COX enzyme. There's 1 and 2. One um, it is involved in the cryoprotectant of your stomach. In other words, so if you take a lot of aspirin, you can get ulcers because it inhibits the COX-1 enzyme. It also tends to be involved in coagulation of platelets. So if you take aspirin, which inhibits COX-1, it will tend to make platelets less sticky. So people that have a problem with heart disease and so forth will tend to take low-dose aspirin to kind of avoid this clotting. So that's COX-1. That's the good enzyme, okay? And we have agents that inhibit both COX-1 and COX-2. Now, COX-2 is an enzyme that's induced during an inflammatory process. This is the one we want to get rid of. and Celebrex specifically inhibited this enzyme. So I had a product that had high bioavailability when given orally. It had good concentration in the gastrointestinal tract, and it was very specific for inhibiting this enzyme that we wanted to inhibit without inhibiting the good enzyme. And so we kind of arbitrarily figured out a dose based on body mass from humans, the human dose in birds. We treated this bird, and I presented this at the AV, I think it was around 2000, 2001. And I actually showed radiographs of this bird. And then 6 months later, you can see how the dilated proventriculus actually shrunk down with this Celebrex therapy. So that's how we ended up with Celebrex. And Celebrex was really good at inhibiting this ganglioneuritis that was occurring around these birds. And so we went on Celebrex orally, and it worked. Well, we had a pretty conservative dose on that. And my colleague, Dr. Susan Oros, had this Amazon parrot that had this cortical blindness. And again, its eyes were sensitive, the retina sensed light, the iris would constrict, but the bird up here in the upper brain couldn't form the image. And there's a problem. There's a condition called the blood-brain barrier, and what that does is it protects the brain from things that are circulating in the, in the blood system, and so in order to cross that, Susan found that she had to increase the dose of Celebrex up to 60 milligrams per kilogram. This is 3 to 6 times the normal dose we were using. And sure enough, that bird regained its sight with that treatment. Okay. And we got to remember, though, that in birds that have ganglioneuritis, that transport through the digestive tract and absorption from the digestive tract is not normal. And between that and the blood-brain barrier, I think that's why she had to use this high dose in order to get this bird to respond in the central nervous system to the Celebrex. We get a lot of veterinarians that use meloxicam because they're doing birds, but they're also doing dogs and cats, and they always have meloxicam, this product, on the shelf. Okay, well, this isn't an oral suspension. We're going to give it to this bird. The problem with meloxicam, it's a COX-2 selective inhibitor. It's not a pure COX-2 inhibitor. So what that means, it inhibits some of the COX-1, the good enzyme that we don't want to inhibit. Additionally, meloxicam was formulated for the digestive tract of the dog and cat. The digestive tract of the bird is much more acidic. The pH is different. And so we don't get really good absorption of meloxicam from the GI tract in birds. So Celebrex is far superior to meloxicam. And high doses of meloxicam, you could probably get some inadvertent side effects by inhibiting the COX-1 enzyme. There's actually one paper that tried to prove that we're on the wrong track in treating birds with Celebrex, and they used meloxicam, and they concluded that it worsened the symptoms in cockatiels that were infected, artificially induced ganglioneuritis. And I don't know what they did. They didn't use an appropriate dose of meloxicam, and I think this paper stands out there like a red herring because we've got veterinarians all over the country that use Celebrex and meloxicam to good effect. Okay, well, since then we've got this product came out on the market called Onsior, robenacoxib. It's a pure COX-2 inhibitor, just like Celebrex. The nice thing about it is we can give it intramuscularly, so we bypass the transport and absorption from the gastrointestinal tract, and and that way we know the bird's getting it and it's going to work. The nice thing about it, we don't have to give it twice a day like we do the Celebrex orally. We can give it by injection in the muscle once every 5 to 7 days, and it works fantastic. And by far right now, this is probably the product that we use the most of, most frequently. Once we get birds to respond, and if we need to follow through with the longer term treatment, we may go to oral Celebrex, and we may do that once a day. But generally, we'll use Onsior once every 5 to 7 days for 3 to 6 weeks of treatment. And the vast majority of birds that are showing ganglioneuritis will respond positively to that. Most birds at that point in time, we stop it. Most birds will go on and be completely normal. Some will relapse. If they relapse, we start them back on another 3 to 6 weeks treatment, and most of those birds will clear up and be normal after that. We do have a few birds, and it's not many, that need to be on Onsior on a more regular basis. I can tell you this from testing, from running clinical chemistry profiles, checking liver function, kidney function. Birds that have been on robenacoxib long-term show no adverse effects on their organ function. And I've got one macaw that when we stop it, within 2 to 3 weeks will relapse, start regurgitating, start not, refusing to eat and so forth. This bird's been on robenacoxib for probably 3 years now, with only very short periods of time where it was not on it. The bird is completely normal. And, some will question, well, how can you justify long-term treatment with this? Well, I can say this. We checked this bird's chemistry. We know its liver and kidney function remain normal. And the alternative to not treat this bird is this bird will go downhill and die of ganglioneuritis. And this bird has been doing very well at this individual's home for well over 3 years now. So a very, very safe and a very, very effective treatment. And in my recommendation, the only treatment that should be given to birds with this until you get the point where the digestive tract is functioning normally. And if you need some longer term therapy, you can go to the oral Celebrex treatment, but meloxicam I do not recommend at all. So the bottom line in some closing remarks is that a significant percentage of the avian population is infected with avian Bornavirus. If you have over 3 birds in your collection, chances are you have avian Bornavirus in one of those birds. A continuum of clinical disease exists of ganglioneuritis. We can see very mild signs to very severe signs. The effectiveness of treatment really depends on the location of the lesions, how severe the lesions are at the time treatment is instituted. Okay. In other words, the sooner you get birds that are showing neurologic signs that can be related to ganglioneuritis, the sooner you get them on treatment, the better they will respond, and the better your chances of that bird reverting back to a normal status. Okay. The wasting bird, the PDD bird, like we saw early on, represents the extreme case of this condition. I see very, very few birds in this condition nowadays, whereas around 2000, we were seeing it pretty regularly. So I think we're identifying this condition more early on in birds, and we're getting them on effective treatment. So the wasted bird, the PDD bird, that's, skin and bones, we just don't see. And again, another point that you have to take from this, if you take anything from this presentation, the majority of ABV positive birds do not and will not show clinical disease in their lifetime. The majority, and I'm saying well over 90% of these birds will remain normal. They do not serve as a source of infection under normal circumstances for other birds in your aviary. We know that Bornavirus fluctuates between dormancy and activation. Periods of activation appear to be triggered by stress, immunosuppression, the breeding season and so forth, elevated reproductive status. Again, we see a lot of birds in a late winter and early spring that'll show up with ganglioneuritis signs. We treat them, they're normal until the next late winter, early spring comes along. We'll use things like progesterone injections, Lupron injections, and the desloratadine implants. It's really unfortunate we can only get the single desloratadine implants here in the U.S. These will work anywhere from, I've seen them as short as 2 months to as long as maybe 78 months in the average citizen bird, whereas the double implants, which are available in Australia, I've seen them last as long as 3 years in birds. So they don't allow them into the U.S. because they're afraid that people are going to use the double implants in dogs, and they're not approved for that species. And for us to get the double implants into the US, there's a lot of hoops to go through. I used to get them. A new company took over in Australia selling them, and it is almost impossible for me to get the implants. I'm still working on it though. But if we can get the double implants, that's what you want to go with in these birds that are positive, especially birds that tend to activate during the breeding season. And we do have some veterinarians that have tried 2 single implants in a bird, and I can tell you they are not as effective as 1 double implant. They don't last as long, and they don't work as strong as a double implant will. So, well, that may be a possibility. It's expensive, and you're not going to gain a whole lot by doing a double implant rather than 1 implant. And even if you got to come back 2 to 3 months later and do another, you'll have better success doing single implants that way than trying to double up on them. Okay. We know that the disease that we see with Bornavirus is called avian ganglioneuritis. It is induced by an autoimmune mechanism. We know it can be induced by other infections, either directly, such as paramyxovirus or chlamydia, that impact the nerves and expose the nerve gangliosides, or by what we term that molecular mimicry, where we can get a sensitized immune system against, say, a bacteria, Campylobacter in the digestive tract, that will then cross-react with certain gangliosides in the bird's body, will cause a ganglioneuritis, and then the corresponding disease signs that we see. So disease can occur with or without the presence of avian bornavirus. So avian bornavirus really isn't this big evil virus that's out there that's we need to really be so concerned about. I think observing our birds closely for neurologic signs that, or any clinical signs that can be attributable to a neurologic origin, you need to get them tested. The AGA test is probably the best test to do that, to confirm that, yes, indeed, we have a ganglioneuritis occurring. So with that, I'm going to say thank you to Anne and Phoenix Landing for hosting this session, and to all of you who have attended. I hope I've explained things a little bit better to you. And I guess, Anne, we're going to take some questions.
Oh, yeah.
Hello.
Hi. Thank
Oh
you,
my,
Dr.
here we
Bob.
go.
Thank you. There's definitely a lot of questions, and
Well, I hope I explained some of them at least.
oh, absolutely, absolutely. And before we go on, for my behavior colleagues, IAABC, who may be here today, your code word is nerves. Just email us at contact@phoenixlanding.org, and we'll give you the code for your CEUs. Yes, Dr. Bob. So thank you for.
I want to mention something, okay? I
Yes.
do have a little FAQ, frequently asked question sheet. I will email to you, and then you can distribute it to those who are,
Okay.
so desire. And I'll do that as soon as we close here today. Okay.
Yes, what he's talking about is a handout for clients and a more easy to digest information, because it's a complicated issue and so many veterinarians have to explain it to their clients. So our goal here was to get something more digestible for them to be able to hand out to their clients. So we'll try to.
I hope it was, and this
Yeah.
this fact sheet is going to be a work in progress. And if there's anything in there that you say, hey, you need to, address this, let me know. We're going to modify this thing as time goes on. Okay.
Sounds great. We'll put it adjunct to the recording on our website, and we can update it anytime you have new discoveries, because One thing you did not say is that your lab is the only one with a patent to test for ganglioneuritis.
Well,
So
it's Dr. Rossi in Italy actually has the patent, and we licensed it from the
there you
University of
go.
Camerino. Yes.
So if any other lab in the US says that they've tested for it, they could only do that if they sent the sample to you. Do I have that right?
That is correct. And we do have one lab that advertises, but they send all their testing to us.
Right.
Not, they don't send all their PCRs to us because they're using a quote discount lab that we've had a lot of feedback on, where people thought we did the testing and it didn't correspond with what they saw clinically, and we had to say no, we actually did not do that, but um, so be it.
If
But they
you
do
want
send
to
us.
stop sharing your screen, then your desktop will go away.
Okay, okay, here's share. Let me see. Share. I don't know how to stop it.
Yeah, you want to stop sharing.
Well, I'm hitting share here, but it doesn't give me an option to stop sharing. How do I do that, Ann?
The same green button should probably,
Oh, at the top, maybe. Let me see here.
wherever that might be on your
I
screen.
see something here that you are screen sharing. Did that stop it?
No.
Oh my gosh. How do I
Huh?
do this? Your
Or
screen.
you could go back to it and it would leave it on. I fear you didn't need your desktop open to the world there.
I'll just do that.
There
How's
you
that?
go. Perfect.
Is that
Okay,
okay? All
that's
right.
great. All right.
Okay.
So my first question, thank you, first of all, for clarifying unequivocally that Bornavirus comes largely through the egg. Birds are born with it. They don't necessarily die from it. And most of them are not symptomatic.
Thank
But
you.
so is it possible for birds to be born with ganglioneuritis, with already damaged nerves or myelin?
No, no, because when birds hatch out, their immune system is not totally competent. This is an autoimmune disease. We do not see disease occurring until approximately around the time these birds are being weaned. That's when their immune system becomes competent. And in the Kias, that was a perfect example, because they were normal as they started to wean, then we started to see the neurologic signs. So no, they're not going to hatch out with it. And actually, the eggs that we tested that did not hatch, we ran histopathology on those chicks that were in the egg. Not a single one of them showed any evidence of ganglioneuritis. So it's an autoimmune disease, requires an intact, functional immune system. We don't get that until about the time of weaning in pet birds.
Okay. So birds being born with the Bornavirus. No, I'm going to skip that. We don't want to go into Bornavirus. Okay. So if both parents have ganglioneuritis and they're having chicks, what are the odds are that the chicks are going to get ganglioneuritis? In other words, do we want to staunch that kind of breeding somehow to get ahead of this disease?
Well, in the Kias, I use it as an example. Both parents were positive for Bornavirus. When we tested the patellin membrane, 22 out of 23 of those eggs were positive for Bornavirus. So pretty good chance that the parents are positive, the chick's going to be positive.
Right, for Bornavirus, but for
Right.
ganglioneuritis, is it the same?
No, because ganglioneuritis is the condition that occurs when the immune system reacts with nerve ganglia. And a lot of those birds, if we don't get exposed nerve gangliosides, we're not going to get the disease. The virus, as it travels up the nerves through the bird, it can't expose these gangliosides and we can get the disease that way. But the virus itself does not actually cause the inflammation. Okay. It's exposing the gangliosides as the virus travels through these nerves that exposes the gangliosides that the host recognizes its form and mounts an immune response. And that immune response causes the inflammation and damage to the nerves. The virus does not damage the nerves. It's a non-cytopathic virus. In other words, you culture that virus in a dish in, like, say, kidney cells, it will not kill those cells. It's non-cytopathic.
When you say the virus, are you talking about Bornavirus or
Yes.
ganglioneuritis? Oh, okay. I'm talking
Born
only about ganglioneuritis so that we don't confuse it with Bornavirus. So if both
okay.
parents are ganglioneuritis positive.
No, both parents were clinically normal. They were
No,
Bornavirus positive.
if both parents are ganglioneuritis positive and they have chicks, are they passing on a weak immune system, a weak nervous system to the chicks?
We don't know.
We don't know.
Because it's pretty unlikely if both parents are clinically diseased from ganglioneuritis that they're going to have enough reserve capacity to produce eggs and viable chicks. And it's hard to say. I mean, they may pass maternal antibody against certain neurogangliosides that can affect the nerves of the chick, but that's just in theory. That's never been proven. Never been proven.
Because I know there's some questions, that are concerned about some aviaries where they know there's positive ganglioneuritis and they're still breeding. I don't think there's anything we can do about Bornavirus. It's so rampant. I mean, we can't stop that from being continued at this point.
The only way you're going to stop it is from not breeding birds.
Yeah, period. Right.
Yeah,
exactly.
Yeah.
Exactly. Several people have wanted to know if you offer consultations. I'm sure you already do that for veterinarians, but for individuals who are looking for help, who may
I
not
try
be
to
near
I try to answer emails as often as I can. That's the
Can they
best
hire
way.
you though as a consultant?
No, I don't charge people for that.
Oh,
I mean,
okay.
if they can send me an email when I get a chance, I'll get to it. By phone is really difficult
I
because
know
we're
how
so
busy
busy.
you are. Yeah.
Yeah, but I mean, I could at 11 o'clock at night get on the computer and answer an email or
Okay.
something.
You
So
may be inundated. Okay.
email is the best.
Okay. I hear you.
Or email the lab, email VMD, that works too.
Okay. Somebody wanted to know if naltrexone is a viable treatment. Naltrexone, did I say that right?
Yeah, I don't think so. I mean, I don't know if it's very effective against the virus. They've tried a lot of different antiviral agents, but this virus hides out the cell nucleus, very similar to herpes virus. And
when you say the virus, are you talking about Bornavirus again?
Bornavirus,
Okay.
Bornavirus, yeah.
I'm so focused on ganglioneuritis. So
ganglioneuritis is a condition. Bornavirus is a virus.
Yes.
Okay.
Okay. Gotcha.
So,
a condition. Okay.
So Bornavirus tends to hang out in the cell nucleus and it causes these persistent infections. It's pretty much isolated from different antiviral agents, kind of like herpes is, although, we do have some agents that if the virus is active can kind of drive it back into the cell and make it go inactive, but you'll never, at least to this point in time, nothing has been shown to totally inhibit or eliminate avian Bornavirus. I just don't think it's going to happen. And while they've shown some of these agents are effective in the petri dish in the laboratory, nobody's really shown any effectiveness of any of these other agents against the avian Bornavirus in a bird.
This is somebody with ganglioneuritis who wanted to know if naltrexone. So I'm sorry, I try to be very careful keeping them separate. So people.
Onsior is the best treatment for that bird.
Onsior is the best,
hands down.
What if Onsior is not working? I've had several questions about that too. If Onsior,
well,
is having it.
One of two things: either they're not using a high enough dose or not treating them long enough, or they've got another condition that is mimicking ganglioneuritis, but it's caused by something else, like, for instance, toxicity. We've been screening some birds through a lab up in New England, and it's amazing what we're finding as far as elevated levels of things like copper and other agents like that. And Scott Eccles actually identified this lab and has him doing this testing, and it's so much better than just testing for zinc and lead. So we know things like toxicities can induce this, other viruses can. I would say toxicities and sometimes some fungal diseases can mimic this, but toxicities are probably the biggest differential we have to avian ganglioneuritis.
And like you said, anything that stresses the immune system can cause it to activate.
Well, it
And
could cause it to flare up, yeah, but
yeah,
there may be another underlying cause going on, like toxicity or fungal infection
right.
and so forth. So,
One of the things I've learned from you over the years that's been so helpful is that if macaw, who can be when they have ganglioneuritis, has that whole digestive system issue, and the poop gets so smelly, and it's usually the result can be Clostridium because their immune system's down. And you have
sure,
this great remedy for that. First of all, a lot of veterinarians don't know how to properly test for the Clostridium, but I don't know if you want to, this is the right forum
sure.
to talk
That's
about.
no, that's a good thing to talk about, and thanks for bringing that up. Um, we get this abnormal transit of ingesta through the digestive tract. And Clostridium is an organism. I mean, it's everywhere in the environment. It's in dirt and dust, and we see birds that tend to dunk their food in a water bowl. And Clostridium is an anaerobic organism. It grows in low oxygen, so you put food that settles down in the bottom of a water bowl, and it's laying there. It's a perfect medium for this bacteria to grow. And typically, if a bird has Clostridium, the stool's gonna be a little bit loose or runny. It's gonna have this foul odor like old socks and so forth. Best way to test for it is a gram stain. I mean, it's really easy to pick up on a gram stain. Our first treatment for that usually is spectinomycin, because we can put that into drinking water, and spectinomycin is not absorbed from the GI tract. In other words, it doesn't get into the bloodstream. In other words, what you give orally actually works its way through the digestive tract and out into dropping. So it stays right where it needs to be to counter this organism. And if repeatedly it's being used, sometimes we'll get some resistance to it. Azithromycin is another one that we will use. Metronidazole sometimes is a good one to use also. But it's just an anaerobic, opportunistic bacteria that's everywhere. All birds are exposed to it. And when they get this abnormal transit through the GI tract, it tends to overgrow in the lower intestinal tract.
Yes, I can vouch for it being so helpful in many situations. The way we get it is through Spectogard, which is that
Yeah,
liquid medicine, and we
yeah,
just, put it in the water. You don't have to corral the bird or cause it any more stress.
it works really good that way. Yeah, and you can once you smell it, you know what it smells like, and
Right,
you can walk by a bird and say, "Oh, that bird's got Clostridium."
right,
You'd have to run the
right.
gram stain.
It's so helpful. I encourage everybody to pay attention to that. I remember Dr. Orris told me, like, 2005, literally 20 years ago, we were having this whole discussion about Bornavirus and all the fear that was out about it. And I asked her if we needed to be careful about how we adopted birds out if a bird was positive for Bornavirus. And she said, absolutely not, for reasons that you understand, that it's vertical transmission mostly, et cetera. Do we need to be concerned about birds that test positive for ganglioneuritis and putting them in homes?
I think you need to definitely treat those birds.
Even if
Um,
they're not symptomatic?
well, if they have ganglioneuritis, they're symptomatic in one way or another, maybe mild, but they're
Oh,
gonna
really? You can
pretty much.
oh, wow. Well, Phoenix is
Oh,
positive and not symptomatic right now, so
well,
pay for that.
but that bird was treated, right?
For a bacterial infection, yeah, that probably was the induced reason, right?
Well, we can have residual antibody that persists for a longer period of time. If a bird is clinical with ganglioneuritis, you need to treat it. If they're
If they're
non-clinical,
clinical, right, right.
right. If they're non-clinical, do they pose a threat to other birds? Probably not, because Bornavirus really is not easily transmitted from bird to bird. any of
That's
these,
assuming it's Bornavirus. I mean, if it's
right, any of these,
ganglioneuritis, right?
and any of these other agents that can produce it, like paramyxovirus can be more easily transmitted, chlamydia can be more easily transmitted, mycoplasma can be more easily transmitted. So if a bird is clinical with ganglioneuritis, I would suggest they be isolated until they're treated and normal, and then they can be put back in with the other birds.
They can be put back in. Okay. So
Yeah.
if they test positive, they're positive for life at that point for ganglioneuritis, not Bornavirus.
No, they're not, because we
Oh,
have watched birds that have elevated AGA levels that have been treated and watched over time, those levels will decline and go back to normal if the bird remains normal.
so
So
I
yeah,
should send in another sample for Phoenix and see if it's back to normal since
I would.
behavior is currently not normal. Okay.
Yeah, yeah,
So it's not like Bornavirus that they have for life. It's a condition basically that's triggered by something.
right, exactly.
Okay. So once
Yes.
nerve ending is affected and damaged, doesn't it stay damaged and
Not
vulnerable?
necessarily, because we can get inflammation that interferes with nerve function, but doesn't necessarily destroy the nerve itself. And that's where the effectiveness of treatment really depends on the severity of the lesions at the time treatment is instituted as to how much resolution you're going to see. Birds that are mild or more, acutely or recently affected tend to respond much better than birds that are chronic or more, the wasted away birds. They're hard to, I mean, you can stabilize them, but you're not going to get them back to normal.
So if they were positive, they were treated, they're now back to normal. Are they still more vulnerable at that point, probably? Or is it just a matter of what's in the environment?
It really depends on their immune system and what triggered the ganglioneuritis to begin with.
Mm-hmm.
They may have a propensity to develop it again. I think birds that we see with ganglio that we treat, we always are on the watch for the signs to recur. Some of them will recur, within months after you stop treatment. Others will not ever recur again. So it just
On wood,
it's
yeah.
so variable. It really depends.
Right, right. I know this is not the topic of the day, but I'm just curious what you think about the current situation with avian flu and what it means for our birds, either going outside or not going outside or.
Well, the influenza virus is pretty contagious among birds. Um, we've seen that this current strain, the H5N1s, the strain that's circulating, has adapted to some mammalian species. I know of no citizen birds that have been reported with it, and with a lot of the outside aviaries that we have in Florida and California, I would think if these citizen birds were real susceptible to it, that we may have seen some cases already. But again, it's going to require contact between migrating waterfowl, which are the main vector of this, and these citizen birds. So your birds that are indoors, and if you take them outside, I mean, they're going to be fine. They're not going to be exposed to it. I really don't expect to see it in the pet birds that we see here.
So there's still more benefit in getting fresh air and sunshine than there is in the concern.
Oh, I would think, yeah.
Okay.
Unless you're putting
Benefit analysis here.
unless you're
Yeah.
putting your macaw out there with a group of Canadian geese or
And the
something,
geese, yeah, they are migrating. Yeah.
Well, they used to be real migratory, but we have them here year round now. I mean, they
Uh-huh.
just stick around, and there's so many of them. It's just they've really increased.
Right.
Yeah, but it's going to be a problem, and I'm not sure if our current approach on, if you get it in a group of a large poultry population, they just kill all those birds.
Yeah.
And that seems, I'm really wondering if we might be better off to find birds that recover or do not get diseased in that population. They may have some type of innate immunity against it, and those are the birds we need to perpetuate in these
Quakers, they seem to
yeah,
be immune to everything except
Quakers, yeah,
cancer.
Quakers,
Yeah.
yeah,
Yeah.
well, that and fatty liver disease.
Yeah, that's true. I'm curious, why do a lot of these ganglioneuritis or Bornavirus studies use cockatiels? It seems so irrelevant.
That has been a big criticism of mine because um, I don't think, I mean, I've seen it in cockatiels, but the cockatiels I've seen it in were ones that have been vaccinated against their ganglioside proteins. Um, I have not seen it in any pet cockatiels. So I can say on one hand, the number of cockatiels I've seen ganglioneuritis in, I can count on one hand. And so we're using a species that is pretty resistant to the disease in these experimental studies.
Exactly.
Plus, the other thing that's happening, and this is another criticism I have, is they're blasting these birds with such high levels of virus, levels that they normally would not be exposed to in a natural environment. So it really is an abnormal type of um, test that we're exposing these birds to. So um, while we can draw some conclusions, we need to be real cautious about the conclusions we draw from these studies.
Hmm. If you had to answer this question, yes or no, would you say that ganglioneuritis is contagious or not contagious? It's an
No,
individual disease, not a one that
no,
passes, right?
right, no, it is not contagious from bird to bird.
Because it's that individual bird's immune system that either is subject to something that attacks the nerves or not, basically.
That is true.
Is that the
The
bottom
mid,
line?
the
Mhm.
majority of these cases, I will say the majority of the cases are caused by avian Bornavirus, which we know is not highly contagious from bird to bird, okay? But these
Because they were
other,
born with Bornavirus and it
right,
activates for some reason.
the other cases that we see that may be due to, say, Campylobacter, for instance, in the gut, um, that's going to require a bird living in the same enclosure with another bird that has a strain of Campylobacter that it gets infected with, that its immune system mounts an immune response to that cross-reacts with the nerve gangliosides. So yes,
Mhm.
we can have conditions like Campylobacter, maybe paramyxovirus, which is not very common in pet birds, or chlamydia, which is pretty rare, but it can induce some ganglioneuritis. So these other infectious cases, which comprise the minority, this very small section of birds that show ganglioneuritis without the presence of Bornavirus, they're pretty rare. I mean, it does occur, but it's not real common. So I'd have to say ganglioneuritis is not very contagious among birds.
Makes sense to me.
Okay.
And I always hear Dr. Ora's in the back of my mind. She always says that when the hormones go up, the immune system goes down. So everybody that's overstimulating their birds and making them sexy, that does not help. That can lead to that kind of immune suppression, right?
Exactly. Yes.
So
So true. So
behavior
true.
people out there, another reason to tell your clients that it has the downside of sexy behavior. Yeah. I know in past talks on this, you've mentioned gabapentin and how it's not necessarily effective for birds in general, but especially for ganglioneuritis. Is that
The problem with gabapentin, it's really not an anti-inflammatory like Celebrex or Oncio or Robenacox. Um, it does help with quote nerve pain, so to speak. The problem with pet birds is that, and Dr. Oros has really alludes to this, is that the dosing that's been reported is really not that effective. I have found that at the normal reported dosages, it really doesn't do anything in my patients. You've
mm,
got to really increase the dose to get any effect.
mm,
And even then, you're better off treating the inflammation of the nerves rather than just making it so that the bird doesn't, quote, feel those nerves, so to speak.
right?
You
It
know,
seems to me that's only benefit is when a bird is recovering from surgery or something, and you want them to be kind of low key.
but
Um,
even then, Onsior is a really good
ah,
anti-inflammatory and pain reliever, and you give one injection, it's going to last 5 to 7 days. So we
ah,
like that.
So you use that after other kinds of surgery? What a
Yeah,
good idea.
yeah.
Okay. I don't think it's commonly stocked, at least with a lot of the veterinarians we work with. They don't often have it in their clinics. Does it have a short shelf life?
No, no, it really doesn't.
So
And we buy
veterinarians
it.
out there,
We go,
stock up.
we buy it. I mean, we buy so many bottles of that. It's crazy. I think probably a lot of them have meloxicam because they're doing dogs and cats too, and they go to that.
Yeah.
And it will work, but it's not as good as Celebrex or Onseal.
Yeah. And I took to heart what you said today about it inhibiting the good COX inhibitor. I don't
Yeah,
understand all the
yeah.
COX inhibitor world, but
Well,
I don't
there's
want to.
COX-1 is the good, COX-2 is the bad.
Yeah.
COX-2 occurs with the ganglioneuritis. COX-1 maintains the cryoprotectant in your GI tract and it keeps your platelets from sticking together and so forth, you know. You inhibit that and, you know, I mean, it helps your blood to clot. You inhibit that and you run into problems. We don't see so many problems like side effects with meloxicam, but you know, it can occur. And the other products are so much more superior that um, I don't know why you would use meloxicam, other than
Mm.
the fact that you've got it on the shelf and it's easy to reach for. I mean, order the Onsior in or order Celebrex in. Celebrex now, generics are out there and, you know, it's very reasonably priced. So cost is not a factor. Onsior
Can
is
you
gone
even
up.
get the over the counter at the human pharmacy Celebrex version?
Oh, yeah. Yeah,
Okay.
you can prescribe
So
it.
the person,
Absolutely.
okay, so that would make it easier for people to get it everywhere if their veterinarian prescribed it that way.
Exactly.
Okay.
Yes.
And is that a dosage you can tell us now, or is that more precise?
I like to use 20 milligrams per kilogram orally twice a day. And in cases like the cortical blindness that Dr. Oros had,
Mhm.
30 to
30
40
to
milligrams.
60.
Yeah, well, I'd go 30 to 40 twice a day. So you're getting 60 to 80 milligrams per day.
Okay. And that gets compounded for birds in a liquid form, or?
Yeah, you'd have to compound it into like a suspension, but
Okay.
that's easy to do. We do it here all the time. So
Well, that sounds great. I mean, I haven't really heard that before. I don't think that sounds like a very manageable way to get help. So great,
it is, but again, Onsior is our go-to.
better.
I have so much luck with Onsior. That's the first drug I'd reach for. Like I say, we get bottles in here. We order it, you know, 2, 3, 4 at a time, and it started off around $100 a bottle. I think it's about 170 a bottle now. So it's gone
But it's
up,
an
but
injection, right? And a lot of people don't
it's
feel comfortable giving an injection.
I
So,
have many clients that when we teach them how to do an
right.
injection in the breast muscle, will take it home and do their birds and do very well with it. It's
Yeah.
not a problem. We use these little insulin needles. They're tiny, they're short, and you do it in alongside the sternum. You are not going to go too deep. If you go too deep, you'll hit the keel bone. You're not going to go into the heart or liver or intestines or anything. Because you got that big plate that's protecting them. So I have a lot of clients that do Onsior injections and they like it because it beats going orally twice a day, especially catching up a wild cockatoo or something twice a day. Catch them up once a week and give them an injection. It's so much easier.
I was gonna say, if you can't catch them up, then giving it orally on a Nutriberry or something can be an alternative, I
Yeah,
guess.
it is. And you
Does
know,
it have a taste, the Celebrex?
depends how you compound it. I've never tasted it, but we usually put flavoring and the birds will take it pretty well.
Uh-huh. Somebody wants to know if ganglioneuritis is similar to Merrick's in chickens.
Um, No, not really, because Merrick's disease is an infiltrate where, I mean, that's a really good question, but that's a disease where we get lymphocytes that actually infiltrate the tissues. They go into the tissues. They cause some neoplastic type growths that occur in the nerves. So, um, in fact, commonly we'll see lesions in the iris tissue of the eye will change color to like this pale gray color. And that's because of all the lymphocytes that have infiltrated into that tissue. So um, I'd say in a way it's similar because it attacks nerves, but it's also pretty dissimilar also. So very good question.
Mm-hmm. Somebody was asking about my dog, which I think is great because that's how I detected things with Phoenix. But most people here don't know that that is a lab with swabs and DNA sequencing, and it's a great way to detect fungus and bacteria that may not be caught up in any other way. So her question was, doing a my dog assay, help diagnose a problem? And I would say yes, wouldn't you?
Well, it'll help from the standpoint if you have Campylobacter in the digestive tract, for instance. My dog uses the Luminous system for sequencing, which only detects sequences that 16S RNA sequence of bacteria. So it will not detect viruses, only bacteria and fungi.
Right.
There's Oxford Nanopore, which we're looking into, but it's pretty expensive endeavor. Will also determine viruses, and I think that's going to be a very big development. The My Dog is not cheap. I think right now it's about $175 my cost to send a sample to them. And I think if they're starting to use the nanopore sequencing, which I was at the University of Belgium looking at that in Ghent, and we're still in talks with them to offer this. The bottom line was it was very expensive, and we basically would have to run it for over a year before we even broke even on it. So I'm not sure if that type of service that would detect viruses like Bornavirus and that is going to offer much more over, say, just running a PCR for avian Bornavirus, which is
But the question
much
was
less.
my dog for bacteria and fungus. Would
Yeah,
that be helpful?
it's really
Yes.
good.
Yeah,
The problem we run into with that is it identifies so much in the sample,
it
and
does.
you gotta look at the percentages because
Right.
you'll have like 85% of this and 10% of this, and then the other 5% is going to be, you know, all
Stuff that
these
doesn't
other
matter. Right.
extraneous organisms that you never even heard of before.
Right.
So yeah, you've got to have some background knowledge to interpret the results properly.
That's true. That's
Yeah,
true.
but it's good, like from a bacterial standpoint and a
Hmm.
fungal standpoint. I mean, we used it in cases where we got some infection and we're not sure the sensitivity of these organisms and what's there. I think it's a little more sensitive than routine bacterial culture and sensitivity. And reason being is most of the labs that do cultures and sensitivities are doing human samples or dog and cat samples. So they're incubating them at 98.6 degrees Fahrenheit, whereas the bird's body temperature is, 5 to,
More.
7 degrees higher. And you have to incubate avian samples at that higher temperature, or at the low temperature, you may get organisms that overgrow the organisms that are pathogenic or meaningful in that avian sample. And that's the problem with cultures and sensitivities for birds. So you really have to find a dedicated lab that cultures these things at avian body temperature.
Hmm. That's why
Plus,
my dog, because the DNA sequencing is really fast and they're not sitting
yeah,
around.
exactly. Yeah. Yeah. Janina does a great job out there. We've talked many times. In fact, when I saw her doing this for dogs and cats, I encouraged her to
Yeah, you were one of
reach
the
out,
first. Right.
reach out to birds and exotic veterinarians, and now that's a big part of her practice. So yeah,
Exactly.
yeah, she's doing
Um,
very well.
I know we've taken up a lot of your time. I have
No, it's fine.
one last question. So we've done some just curiosity testing, just like we did with Bornavirus way back when, which seemed to
Yeah.
correlate more with feather picking than anything else in our experience. But so if a bird, so we've done some curiosity testing, and there are a lot of birds who've come back positive for ganglioneuritis, but they show no symptoms whatsoever. So what does that mean?
That means that they've got detectable antibodies circulating. Okay. But it hasn't been enough. And you know, to have detectable antibody, you have sensitized B lymphocytes. The T cells are the killer type cells that actually do more damage than the antibody. The antibodies get attached to the ganglioside, and then that activates macrophages and the complement cascade that will then damage that nerve membrane. But again, most of those gangliosides are sequestered within that membrane. And if the antibodies can't get to them, they don't cause a problem. So it's not uncommon to have birds that'll have a detectable level, but yet they're non-clinical. And those type of birds, you just got to watch them close. You know that I need to pay closer attention if I see any signs, clinical disease that can be attributable to, you know, a nervous system, you know, etiology, then I need to get that bird checked or treated. You know, get them on treatment right away. And then if it's ganglioneuritis, they will resolve pretty quickly.
And then when you do that test, there's like a range, right? With a number that
Yeah.
generated,
Yes.
but that number doesn't always come back on the results. Is that something people need to ask you for more specifically, or is that something that could be included?
We're going to start reporting a quote titer, okay, a number.
Oh,
Um,
good.
yeah, we've gone through in the last 6 months, going to a customer portal system and upgrading the whole computer system
I'm
and
still learning
everything.
it. Yes.
Yeah,
Yeah.
well, we're still learning it
It's
too. I mean,
new. Yeah. Yeah.
it took us, well, since last June, we've been working on it. Once we get that up, and there was a problem with reporting actual numbers versus a positive or negative test result. So they've had to redesign their software in there. And so eventually, I would say in the near future, we're going to be reporting numbers. And that's going to be important because, you know, we kind of use arbitrarily 200. Okay, birds above 200 are considered positive, birds below 200 negative. But what do you do with a bird that, you know, comes out at 198 or something, you know? It's
Exactly.
definitely above normal. It's is it going up? Is it going down? And that's where the numbers will help. And I
Right.
actually talked to Brian Spear last week about that, and we're going to be reporting things that way. We do to a couple of veterinarians who've been real helpful in trying to elucidate what these numbers mean, and we report numbers to them. But pretty soon we'll have numbers rather than just a positive or negative.
I'm sure you can't answer this definitively, but is there a certain number where you start to see, generally where you start to see symptoms?
I would see it say any bird above like 275 to 300 is going to be clinical.
Okay. Thank
Birds
you.
between 200 and above, birds above 200 can be chronically infected and treated. And then we know when we treat birds over time, the number drops below 200.
Great, which means it's manageable and not
Right, right,
escalating.
right, exactly.
How often do you see birds die from this if they're assuming they're being treated?
Rare.
Great. That's great news.
I have not had a bird die from ganglioneuritis in, I'd say, the last 5 or 6 years that I can even remember. I can't remember. I mean, back around 2000, we had birds, but not recently. No. Mm-mm.
That's wonderful news. And we can start extinguishing the term PDD, which causes everybody to get panicky,
Oh,
you know?
it's that Pandora's box I showed you.
Yeah, yeah. That's why I love the new term, because not all birds show proventricular issues, you know, it's not
Exactly.
appropriate. Right.
The interesting thing is, like in cockatoos that are showing seizures or African grays with seizures, if you run histopathology, some of them will have that lesion in the digestive tract, but they're not showing digestive tract signs, but you can see the
Hmm.
T and B cells infiltrating around the nerves in the GI tract.
What does that mean, that they're just vulnerable and need to be more closely managed somehow, or we don't know?
I
Pandora's
don't know. I
box.
don't know what it means, really. I mean, other than the fact that, yeah, we've got a ganglionitis in the GI tract, but it's not causing clinical disease. The clinical disease in the central nervous system is much more severe.
Mm-hmm. Okay. And birds can test negative for ganglioneuritis, but still be vulnerable to it, I assume?
Yes, we see that in birds whose immune systems are stressed and not totally competent, and they can show disease and have a very low AGA titer. It's not common, but it can occur. And that's why I say the AGA is not 100%, but it is probably the most accurate indicator that we have of ganglioneuritis occurring in that bird.
And Dr. Giacomo Rossi, who's such a gem in Italy,
Oh, he's amazing.
who's kind of got all this moving forward for us. What's he studying now? Is he on the cusp of anything new?
I think I think I last talked to him around a little bit after the holidays. Um, he's a little bit discouraged by some of the other researchers. In particular, we had an event that occurred in Ghent where one of his PhD students presented a paper and was severely criticized by a veterinarian in Switzerland and one in Germany. And to the point that I've been composing a letter to both of these individuals because I think it was very improper to take a new student that way and to criticize them among their peers. It's inappropriate, and they should have pulled them aside and spoken with them and expressed their concerns. Not only that, is that these individuals that made this criticism were totally erroneous in their assumptions, and I need to bring that out. And I know we've been requested to do some cooperative studies with some researchers in Germany, and I've actually held back because of that situation. So I think Giacomo's a little discouraged, but I think eventually we're seeing progress, and eventually, I mean, researchers are catching up. Giacomo's always 2 steps ahead of everybody. It takes time for everyone else to catch up to him.
Right.
So,
which reminds me, that's another last question. He was such a staunch supporter of Civomix, a probiotic in Europe, which he thinks has great assistance for ganglioneuritis, but we can't get it here. Well, he said that next best was Visbiome. Is that sound right?
Yeah, the problem and the thing about Civomix is it was actually began formulation back in the 80s, well before all these, pre and probiotics were, became, quote, the fashion. And there was a lot of work put into it. And he's got studies to show hand feeding Amazon parrots, how they grew better, they had better feed efficiency, they were healthier when that was added.
More like their parents would feed them.
Right, exactly. And the problem we ran into when bringing it to the US was that the FDA didn't know how to address 2 or 3 different bacteria that were in Civomix. They didn't know what to do with them, so they didn't allow them into the US. So to make the other product, they dropped those bacteria out of it. So while it's a good product, it's not quite as good as the Civomix is, and hopefully we'll get that approved. But
is that in the works? Is that in the works? Because I remember Giacomo said that those 2 bacteria were the most important ones. Actually,
they were the most important ones, exactly. And the FDA wouldn't allow it because they didn't know what to think about them. So they said, okay, we just are not going to allow them in. And it's so ridiculous because you could probably culture them here, So
is there any hope for that being processed?
I don't know where they stand, but I'll ask them about it,
I remember we met about it after one of the retreats several years ago, and there was momentum for a while. Yeah.
When I go to Europe, I have to stock up on my supply of Civomix and hope that customs doesn't confiscate it.
I know. I think I took the last of Dr. Orris's stock. Yeah. It's definitely a great product, and I think it would help birds who are clinical. Yeah.
It's so much better than what's out there.
Yeah.
It's real interesting. I was approached by a group, and they wanted to wanted me to demonstrate, they wanted me to do a project with them to demonstrate that by feeding birds their product, they would eliminate Bornavirus. And my head just blew up. I said, I'm not going to get involved with this.
No,
First of all, it's not going to happen.
they were
And
born
two,
with it. Yeah.
yeah, and they want me to say something that's not true. So,
Yeah.
anyway, that's an aside.
Yeah. A lot of competition for resources and fame out there, unfortunately, in the human
So
world.
true, so true.
Yes. Well, I'm sure that those who've stuck it out with us are all people that have really, really got an invested interest in understanding these complicated issues. And I suspect your request for client consults or visits to your clinic. Do you want to say the name of your clinic so people can make an appointment?
It's just my name here in Milford, Ohio. Just look me up.
Okay.
I've got Dr. Cummings, Allison Cummings, who works with me. She's ABVP, avian boarded, and she's doing a great job. I kind of hope she would take over, but she, wants to start a family, doesn't want to run a business. So
The youngins.
we're gonna be here for a while. But again, email, you can share my email. And
So if people look up your clinic online, what do they look for the name?
Bob Dahlhausen, DVM.
Oh, okay. You don't have a hospital name. Okay.
Avian Exotic Animal Medical Center.
Because I think a few people here today are ready to call, make an appointment, and bring their birds to you.
They should just go to support your local veterinarian and just, if you
Have
want to
them call you. Yeah.
have them call me or have them pull blood and we'll run an AGA level. That's all.
Well, I know we had quite a few veterinarians here today and probably in the future too. So I love that you plugged Celebrex and Onsior as something important. To keep on hand.
And Visbiome Vet, because
And Visbiome, yes.
that's the next best thing we have, the Sevomix.
Exactly.
It really, even without lacking those two important bacterium, it's still pretty much superior to, in my opinion, to anything else that's out there.
I think it's gotten pretty well known in the dog and cat world, which it's made for. So probably, guys, you could probably call a dog and cat vet to find it. It's we used to try to buy it wholesale and sell it, but it has a short shelf life, so it's better.
Yes, it does. Well, maybe an individual can buy it direct. Then I don't know.
I don't think so. I think you have to get a prescription or get it
Oh,
through
do
your
you?
veterinarian.
Really?
I'm pretty sure.
Okay.
Yeah.
I'm not sure. Okay.
I'm not sure either. But yeah, I highly recommend it. It's a good product. So, well, thank you, Dr. Dahlhausen. You are a treasure to.
No, you're a treasure, and we appreciate all that you do, not only for the birds, but for the aviculturalists and the veterinarians and hosting these events. I mean, you're a real gem, and we thank you so much. You're great.
Hey, we're all in this together.
All that you do. Thank you for all that you do.
Better for the birds. Yeah.
There you go. There you go.
One step at a time. Well, we need you on the team. That's no doubt.
And we need you too. So, and it's great to see you. You're looking great.
Likewise. I hope to see you soon at the next APV, etc. So,
are you going to Phoenix?
I hope so. We have another echo tour planned for Brazil in September, which I never want to pass up. So anybody out there who's interested in our echo tours, email us and we'll let you know what the next options are. But so I hope to go to Phoenix as well. That's one of the highlights of my years to geek out on all the latest medical stuff. So I can ask you intelligent questions, hopefully. Yeah.
I'm going to join you on one of your echo tours one of these years.
We hope so.
I actually just,
that's a Brazilian green wing right there. Yeah.
There you go.
Yeah.
I just got back from Lapland about 3 weeks ago. We were in Lapland. Amazing. Snow, dog sleds, reindeer. It was the coolest thing. It was really cool.
Sounds wonderful.
I would go back. So anyway, but I hope to see you soon. And thanks again for all that you do. We really appreciate you. And thanks for everybody that attended this webinar.
Yes, you've got some new fans, no doubt about it. So,
thank you so much. Take care,
Ann. Bob. See you.
Thank you. See you soon.
Hey, everybody.
Bye.
Next weekend, join us if you want to for Q&A with Dr. Orris, Rosemary Lowe, Chris Shank, and April 12th, Cassie Molina is back with us to help some advice on how our birds can feel less stress when they see their avian veterinarians for things like this. So that's also very helpful. So I hope to see everybody again very soon. Thanks.
Bye-bye.
Bye.